Proteins are a unique group of biopolymers. They're made up of amino acids. Amino in there, which means that proteins are going to have a nitrogen unit in it. Proteins function as enzymes, which tend to make reactions occur quicker, or they provide structure. So let's take a look at what is in a protein and what happens with proteins. The standard amino acid looks like this. You can see that it says there's 2 carbons, there's a nitrogen, there's 4 hydrogens, there's 2 oxygens, and then we've got this thing called an R group. And the R group is variable. It can range anywhere from a simple hydrogen all the way up to some fairly complex structures that we're going to take a look at. It's called an amino acid. The acid group is COOH, where you've got the carbon double bonded to an oxygen and also bonded to a hydroxyl unit. We've got the amino group, which is the NH2 part of it, hence the name the amino acid. When these come together and they bond together, we get what is called a peptide bond, where you have a carbon double bonded to an oxygen, then bonded to a nitrogen. This is the way that polypeptides are created. Poly means many, peptide means that they've got peptide bonds. So it says we start out polypeptides, we have a series of amino acids. This particular one is glycine. You can see the R group in this particular case is an H. It is going to bond with this particular one. You can see that this R group has a CH2OH on it, and this is serine. And this happens by removing a water. You see the hydrogen from one amino group and the hydroxy from the acid group come together. They are pulled out. We now have a peptide bond in between the two. If we take this and we put another amino acid in, and this one is threonine. Now look at this one. This has 3 carbons. It's bonded to the middle carbon. It's got methyl groups on either ends of that, and it's got a hydrogen in the middle. And again, we're going to take out this water group. We're going to get another amino bond in here. Hence we have the beginning of a protein. You can see it's got various amino acids in it. This particular one has 2 amino bonds in it, so all of a sudden we've got a polypeptide. When we deal with this, we deal with protein structure, and protein structure comes down to 4 different kinds of structure, so to speak. One is the sequence of amino acids, and the one we just created was a glycine serine threonine unit. The secondary structure is basically, if you imagine these polypeptides as containing hundreds to thousands, possibly 100,000 amino acids in it, they get to be rather large molecules. And a large molecule like that is fairly unstable. So what happens is it folds back on itself, or it twists in order to give it some sort of structure so that it'll be less likely to fall apart. And we call this the secondary structure of it. It says the secondary structure, what you get is you can have little twists, you can have little sheets. These fold back on each other. Basically, it gives it more stability in there. If you take tertiary structure, that's where you take the sheets or the twists and you twist and you sheet them on top of this. So it makes a much more complex molecule where everything is much more compact and much more condensed. And we call that a tertiary structure. The quaternary structure is where we take different smaller proteins and we put them together in a larger unit. You can see on this one how many different colors are in here, and each color theoretically is going to represent a separate enzyme on itself, and then all of this together gives you an enzyme that is going to be much more complex. One of the things we talk about as far as proteins go is proteins tend to have a maximum and minimum and optimum where they stay at, and if you take them outside of that, you can actually denature them. What denaturing says is denaturing is where you're going to take the nature out of it. We can denature proteins with a lot of different materials. This one says acid. And acid, basically you're looking at having a pH that is going to be problematic for the protein. And we call this particular process pickling. Jar of tiny sweet midges. I mean, these are really nice, sweet cocktail pickles. There's a lot of different ways that we can pickle material. Again, it's using acid in the form of vinegar. We can use base, and in base, what we're doing is treating the material with lye. This is a process by which we create something called hominy. You know, you live in the South, it's treated corn, where they take corn and they soak the corn in lye and then they dry it down and they use it. They can grind it up, and we call that grits. Here's a couple of cans. One is white hominy, the other one is golden hominy. It depends on whether you use white corn for this or whether or not you use gold corn. If you look at the back of this, one of the interesting things to look at on the label is the amount of sodium. It says sodium is 530 milligrams, or 22%. That's kind of high. That's in part because of the way it's manufactured, and it's basically manufactured by soaking it in lye, and then you rinse it off, but you never really get all the sodium out. You can denature proteins by cooking them. Cooking them, what you do is, well, that's my breakfast one day. Egg cooking over the stove, and what you're doing is denaturing the proteins. When you put the egg in, the egg is a lot of protein, and rather than drying the egg down, what you're actually doing is you're denaturing the protein so they are easier to digest. The amino acids will come apart easier. And you can do it in alcohol, in which case we call it pickling to a certain extent. And here's a jar of brandied peaches. If you look at the original recipe for maraschino cherries, it consisted of using alcohol as well. But in this case, brandied peaches and other foods like this use alcohol. Now, once you denature it, you can't get the original protein back ever again. One of the functions of proteins is that of enzymes. And what an enzyme does is the enzyme basically takes the energy of activation for a reaction and decreases it. Now you can see you've got two graphs, and the two graphs has virtually the same curve. One just has a lower energy of activation than the other, and what you can see in this is the one with the lower energy of activation occurs much more rapidly than the one with the higher energy of activation. So in this particular case, what you get is the reactants come out and they combine much quicker, so you have the process occurring quicker. And with enzymes, things occur up to 1 million to 1 billion times quicker. When we look at amino acids, there is a group that we call essential amino acids. And the essential amino acids are basically those that you must have in your diet. When we start breaking these down, we start looking at them, and we see certain things in some of them. Methionine has a sulfur unit in it, which makes it a little bit more difficult. Tryptophan, phenylalanine, and histidine have ring structures in them, which make them more difficult. And then the other ones, the valine, the threonine, the isoleucine, the leucine, all have much more complex side chains on them. Therefore, these are a little bit more difficult to form, and that's why they're called essential, because you must have them in your diet. A food material that is very good for essential amino acids is something like beans. Beans tend to have a lot of these essential amino acids in them, and that is why beans worldwide make up a tremendous amount of protein for people. Back to proteins. When we look at proteins, proteins are polymers. The monomer is called the amino acid. The polymerized part is called a polypeptide or a protein, and proteins have one of two functions. They either function as enzymes, making reactions occur quicker, or they are structural in nature.